Okay. Yeah, we're gonna before we continue with the material, last time I asked you to download this program called CyberDAC. Uh, do all of you have CyberDAC in your computers? Great. So CyberDAC is a software that lets you connect to your Hoffman account or any other remote host and lets you do file transfer. So here I'm already connected, but if let me share slides from day one. Once you open CyberDAC, let's see, okay, you can go to the open connection option over here. And in that option, you select SFTP protocol. And down here, you just put your Hoffman credentials, username, and the Hoffman 2 domain. Is it visible? Is it, can you guys tell what it says? It's a little blurry, right? Yeah. Oh, no, this is that way. But here you pretty much have your Hoffman2.idr.ucla.edu, and that's for your server, then username and password. Was anybody able to connect? Yeah, great. Okay. So once you have connected to Hoffman, transferring files is pretty straightforward. You can go to this action button and select upload. So I can show you from my end. Okay. So you should have something like this where you're connected and you can see all the folders that you have in your home directory. So for most of you, it's probably just the Unix folder that you created. And here you can go to actions. See, where is upload? Upload. And from here, you can select a file from your local computer. So for example, if I want to upload this PDF, just click upload, allow. Authenticating may take a bit of time, but the actual transfer should be pretty fast. Yeah, here we go. Okay. And now, once it refreshes, here we can see in my home directory, I have the file that I just uploaded. Okay. So this is a very convenient software to upload files, especially if you have to upload multiple files at once. But you can also do transfers from the command line. So let's go to the terminal. Okay. Now we're going to transfer a simple file. So let's first create the file that we want to transfer. Let's do v readme.txt. And in this README, we can type something very simple. Say, for example, intro to Unix command line. Command line. Yeah. Now, do we all remember how to exit the Vim editor? Okay. So first step, escape, colon, WQ, so save and exit. And now we have our README file. Okay. Quick show of hands. Do we all have the README? Okay. Okay, so now we have this file that we want to transfer from our computer to the Hoffman cluster. The command that we're going to use is called scp. And it's similar to the copy or move function that we looked at yesterday. So we just need to type scp source and destination. So scp readme. And now to transfer to Hoffman, the way I'll do it is I'll put my credentials, username at Hoffman2, IDR, ucla.edu. Okay. And so this is just to connect to Hoffman, but then we also need to specify where in Hoffman we want to move it. So we use colon and we put the name of the folder. So I can put it directly like this because the default is your home directory and we have this folder in our home directory. Press enter. It's going to ask me for my password. And here we have a quick progress bar. Should be fairly quick to transfer. Oh, just so you what? Do you get the progress bar, this line? Oh, that's weird. Are you already connected to help me? You're already connected. Okay. My understanding is it should ask you every time, but does it ask for password for everybody? Yeah. Okay. Yeah, I'm guessing because you're already connected. So sometimes when you put your credentials, it allows you to connect again like soon after. Like how long ago did you connect to Topman? Okay, that must be why then. Yeah. Mhm. Where it goes? Yeah. So right now it should be located on the Unix. WS. So the way to check would be to log in and see we actually have the README. Um, so uh-huh. Right, this is my local computer. Yeah, yeah. Um, so right now, this tells you where I am on my computer, right? This is my home directory. I can check what my home directory is. User Giovanni. So here on my Finder, I think you have to go to this your MacBook. No, here. This is my home directory. And here would be the README. Yeah. And here it says intro to command line. Yeah. Mm-hmm. Right. Exactly. Yeah, that's another alternative. Right. Yeah, that's true. So yeah, and that's an important point because somebody asked me yesterday that, for example, if you make a plot on Hoffman 2, you make a PNG or JPEG graph, you can't visualize it, right? Because it's just a terminal. So one way of visualizing would be to use CyberDAC. You can go to your directory, and from here you can open any file just like you would in your local MacBook. Yeah. So, for example, this PDF. So this is right now on Hoffman, right? And this is a PDF file. See, I'm gonna click allow. Yeah, it's a little um, like redundant because now to open it is gonna download it, but yeah, I mean it is a way that you can check stuff. And once you download it, it works like any other file. Mhm. Right, yeah. So this is a file transfer software. That means file transferring means uploading and downloading. There is another option to do this, which is mounting. Mounting, this is also day 3 material, but I can just quickly show you. I have a script here that directly lets me mount Hoffman. So when you mount Hoffman onto your computer, you're not doing file transfer anymore. It's like Hoffman is a folder within your computer. So for example, let me go here. To mount, you use this software called MacFuse. And here you can see now I have all my Hoffman material on this folder. And same thing as before, I can open a PDF on my Acrobat Reader. Yeah. And I can visualize graphs and everything. Oops. Okay. So mounting is better when if you're doing a lot of file transfer. Oh, can I close this? But if you just need to send files just one time, it's also easier to do it with SCP or CyberDAC. It's more straightforward. Okay. Any questions so far? Oh, the Scratch folder. That's a good question. Let me see. Okay, yeah. So it lets you go all the way down to U, Scratch. Let me see, where is G? And now I just find my username. And here I'll have my Scratch folder, yeah. So the way you can tell is that when you click on here and you see your directories, you can see you have the option to go all the way down to the root. So that means you should have access to Scratch. Oh, you Scratch initial and then username. Okay. Any other questions? We're good on file transfer? Great. Okay, so before we log into Huffman, or if you're already logged in, so please log out for now. Remember, you can log out by typing exit, or you can also do Control D, and that will log you out. So now that you're logged out, yesterday I very briefly mentioned the alias command, which is a way to pretty much like a quick access to a command. So instead of typing the full command, you can alias, we can assign it a name. I'm gonna put like hlog equals, and then the command in quotation marks. Okay. So alias, here I selected hlog, but you can select whatever you think is gonna be easier for you. And then followed by equals and the command in quotation marks. Yeah. Yeah. So this is a quick hack. So now you can just type hlog and it asks me for my password to log into Huffman. So now I don't need to type everything. But now there is a problem, right? Because everything is saved in this current session, which means that when you unplug your computer, when you turn it off, and then you reboot it, this alias, this variable is going to get lost, right? So it just works for this one session. So how to make it permanent so that you can use it anytime? This goes back to what I was talking about the first day about environments. So you can define an environmental variable that contains this alias so that it's permanently present. So in your local MacBook, we all have MacBooks. Okay, great. We're going to do this. VI squiggle dash dot bash rc. Oh no, sorry. Squiggle dot bash, let's see. Hold on, give me 1 second. Oh no, bash, sorry. Squiggle dash dot bash profile. Yeah, this is a bit confusing because on Hoffman it's called bash RC, but then on your computer it will just be bash profile. Okay, so before we edit it, what can we say about this? So this means it's located in your home directory. That's the squiggle. Now, I also very briefly mentioned this yesterday, but who remembers what it means when it has a dot in the front? It is a current directory, yeah, but in this case, it's part of the file name, so it has a different meaning. So this is an example of a hidden file. So this is a file that you won't see on your Finder folder, but that you can easily access through the command line. And this is a bash profile. So this is, you can think of it as a settings page. So the place where you define variables for you to use. Now we press enter. And you should all have, probably for most of you, this is completely empty, right? Or does any of you have something? You have something? Do you have this conda thing or do you have other things?
This is like the example.
Mhm. Right. Does somebody have something different? Okay. Right. Anybody? Okay. So, oh yeah. It says what? So yeah, just make sure you have the right spelling. Yeah. You have something different?
Yeah, I think the only difference is they don't do something so underneath that.
Right. Anybody else? You? Okay. Yeah. So whatever you have here already is going to depend on what programs you have installed before. Because some programs that you install on your Mac, they're going to need to modify some environmental variables. So it automatically writes to this file. So that's how you can tell. So in my case, I have this conda initialized because I installed, and conda automatically adds this so that it knows how to manage the different environments. I also have these other lines over here so that this is more of a version control. So sometimes you have the same package in different versions. So you can point it like explicitly. I want to use this specific version over here. But anyway, so a lot of the content in this file gets added automatically by the software that you download and install, but you can also manually add stuff. So here, if you press I for insert, now you can add whatever alias you want. So for example, we talked about hlog. This will be my login alias, SSH jobas at Hoffman. ibre.ucla.edu. No. Make sure you use the same quotation. Okay. And for most purposes, yes. Later today, actually, we're going to find a few cases where you can't. But for the alias, it should be fine, either one. Okay. So now that you have written your alias, and remember, you can use this for anything. So for example, I don't want to type this long command. So I can just do LT equals this. Who remembers what this does? Yeah.
Listing all the files.
Uh-huh.
You want
Yeah, exactly.
file.
Yeah, exactly. Modification time. Yeah, that's exactly the definition. L for long listing, so one file per line, and T means sorted by modification time. Oh yeah, sure. Yeah, that's a good one. Yeah, so this is what I was also mentioning last time. The way I have it configured in Hoffman is that I replace remove with the remove I. So that whenever I delete something, it's gonna ask me, are you sure you want to delete it? Mm-hmm. Yeah, so when you define an alias, you can call it anything, right? And here, this LT is summarizing this command, which is the long listing of file. LS is the listing command. L means long listing, and T means sorted by time. So when I list the files, I'll get the most recently modified first and the older files at the end. Yeah. Okay. So now that we are done writing our aliases, we save it. So escape, colon, WQ, enter. Now, the way we apply these updates is with source. So we replace the VI with source. This command source just means like load this file. So apply the changes. So you only need to run source when you want the changes to be applied to your current session. Because next time you power off and then turn on your computer, it automatically runs this source command every time you turn your computer on. So you only need to do this once after you added a new change. Okay. So now our aliases should be active. I can do LT, and I have the long listing. Let me see. I'm gonna try to delete one of these folders. Okay, I'm gonna try to delete this one. Remove IGB snapshot. Oh, this is a folder. Okay. Never mind. Let me try something else. Okay, I'm gonna try to remove one of my desktop files, one book. Remove desktop books. Okay, so I'm gonna try to remove this, and it asks me, remove this file. I put no. And our login, hlog, and now I can connect to Hotmail. Okay. Oh, wow. I almost delete my pieces. Okay. Sorry. Yeah, sorry. Okay. Are we good? The source command. Yeah. So once you modify all your bash profile file, so far all you've done is just modify a text file. But then if you actually want your computer to implement these changes, you have to run source. And you only need to do it after you added new edits. Because next time when you start a new session, your computer is going to automatically run this command. It's automatically going to read from this bash profile file. Right, exactly. Yeah. The way, sorry. So the way you can think of it is when you have a Microsoft Word file and say you want to save the changes, right? You have your icon. So this could be the way of saving changes at the systems level, right? So that it remembers the changes you made. Yeah.
We're not talking automatically when you're like, you can use not often.
Mhm.
You got to automatically say it, like you do it.
Right. Yeah, that's a great question. It's the same thing. Every time you log into Hoffman, it's going to be a new session. So it's going to forget whatever you define in the previous session. So in Hoffman, we can do the same thing. But like I mentioned, in Hoffman, it's called different, it's called bash RC. And well, here in Hoffman, I have defined a lot of things, but you can see I have defined also a few aliases. It works the same way. They are both Unix based, so you can also define your alias in your Hoffman account. So I would suggest you add this, remove equals remove dash I, just for safety. This is a command we're going to learn about today. This is very useful. And I always misspell it. So I define an alias so that even if I misspell it, I get it right. And you can check your running jobs. And for some reason, I always type my jobs. So yeah, so if you have some consistent mistake that you make when you type command, you can also define an alias. Yeah.
So, your VI, bash profile, that edit that file,
Mhm.
and then the following, right, but
Yeah.
you said the source is like saving it on the system.
Yeah. Maybe that was not the best way of saying it. Because when you do colon WQ, this is saving as text. So whatever you wrote is gonna stay there. Now when I say do source, this is more for your system to implement these changes. Because right now, whatever edits you make, your system doesn't recognize them yet until you load them. So that's the way you can think of source. And this source is gonna get run every time you start a new session. So of course here it doesn't say, but whenever you log in, you're also loading whatever environmental variables you created in your bash RC. So WQ to save text and source, you can think of it also. Yeah, so your command line remembers, I guess. Yeah. Uh-huh. Sorry, can you repeat? Uh-huh. Oh, I see. Yeah, that means you have like a spelling error in the above C. So you have an unmatched proposition. So that means you open potentially, but you didn't place it. Yeah, in your batch of C. So you'll need to reopen. I think it says line 4, right? So just go to line 4. Yeah. Okay. Any other question? Good. So now we talked about alias, file transfer. think we can continue with our material. So for today, we have some more advanced command lines. Today, we're going to learn about these different commands, set, awk, and grep. And regular expression, which is a concept that generally applies to all of these commands as well. These are commands that let you do file editing at a greater scale. So far, we only learned about the vim command. So that means you go into the file and then you can manually edit or add text or remove or whatever you want to do it. But say if you want to modify a file that has 1,000,000 lines and you want to do a systematic change, these are the commands that are going to allow you to do that. Okay, so same format as yesterday. These are the topics we're covering. We already had sort of a very quick summary of day one, but you guys already know how to connect to the command line. We created an alias to do that. This is a quick reminder of some of the commands we learned yesterday. The SSH, exit to log out, PWD, the current working directory, CD to change directory, ls to list files. We also went through some tips, up and down key to browse your command history, tab key for auto completion, clear to clean screen, PW and ls. We talked about the structure of your environment. Here in the slides, you also have this table, which is a summary of the functions that we already looked at. There are a few here that we didn't cover, like gzip and tar. So these are functions that I didn't explain yesterday, but we're going to learn today when we go through the material. Okay, so now we're going to look at these different tools that will allow us to do file manipulation. Okay, so now for our purposes, we're still using, very low memory, very low computing resources. But now we're gonna log into an interactive node so that you get familiar with the commands and how to do that. This is the command that you use. It's called QRSH. And with the -L, you also specify the resources that you are requesting. So in this example, you're requesting for 3 hours of computing time, and you're requesting a RAM memory of 256 megabytes. Which for our applications, that's plenty, but these values also have defaults. So if you don't specify them, you will get by default assigned 2 hours and 1 gigabyte of computer time. Okay, so let's go to our command line. Make sure you are logged in on Huffman. And now we're going to request a computing node. QRSH-L HRT for time. Let's all do 3 hours since our workshop lasts for 3 hours. Comma H data. 256 may not be enough. Let's all just do 1 gigabyte. I think it's easier. Now, depending on how busy the cluster is, it may take longer or shorter time. But usually the rule for less resources, the quicker you get assigned. Hoffman 2 in particular, it has some cutoffs. So for example, if you request 64 gigabytes or more, then you'll definitely have to wait a longer time. So depending on the state, but sometimes you'll have to wait a couple of hours to get 64 gigabytes. Also, if you're requesting a long time, like more than 12 hours, then it's also gonna make you wait some time. Okay, seems that I got assigned a node. Is everybody logged in to the computing now? Still waiting? Yeah, this, yeah, that's a good observation. Yeah, so before, like I mentioned, we're in logging node, but now we're in a computing node, and that's shown here. These messages, these are coming from my bash RC, because in my bash RC, I'm loading these different packages. So it's just telling me that in this new node, I'm also loading these packages over here.
Yeah.
Okay. Great. Okay, everybody logged in? Great. So same idea as before. If you do pwd, you see you are in your home directory. And now what we'll do is let's move to our Unix folder, cd Unix ws. And what we'll do is we're going to download some material. Okay. So how many of you are familiar with GitHub? Okay, some of you. We have some material to work with that was uploaded into the GitHub of one of our collaboratory fellows. And here you have the link. So if you downloaded the slides, you can just copy paste the link into your browser. And if you don't have it handy, you can just Google the words Sergey Nyasep, QC Bio, GitHub, and you should be able to get it. So let me do that. So I'll do Sergey Nyasep. GitHub and Unix. Should be this first link. We go to repositories and should be this one, QC Bio Intro to Unix. Okay. Quick show of hands if you are already in the page. Okay, great. So Unix in itself is its own workshop, literally. But just if you just want to download the materials from one repository, the way you can do it is go to code. And here you'll get an SSH link. You can copy it. Now you can go to your command line and type git clone and paste the link that you copied. Yeah. So this is the GitHub page. You go to code, and here you have the link. Yeah. Oh, you don't get this?
Yeah.
Okay. I see. Oh, all of you, really? Okay. Okay. You can try with the HTTPS link also. Okay, great.
You just paste that link into that.
Yeah, exactly. You copy it, and then again, the command is git clone, and then you paste it. Yep. So whenever you're getting code from or material from GitHub, this is the easiest way of doing it. But there are multiple ways. Another way is to download the zip file. Of course, this will download on your local computer, right? So I'll give you guys some homework now. I want you to download zip and using file transfer the zip to your Hoffman account. Okay, so remember you have cyberduck and you have the scp command to do that. Okay, so it's 2:19. We're gonna take a 10 minutes break plus say 5 minutes to complete the transfer assignment. So we'll continue at 2:35. Okay. So I realized that there might be a few tips that maybe I should share now to make your life easier. One thing that we were already mentioning is opening new tabs, so that you can have Hoffman on one tab and then have your local computer on another one, and like that have multiple tasks going at the same time. In my case, in my terminal, I have this plus sign at the end. That's how you can create a new tab. It's also on the shell, new tab, or just Command T. Now, another pointer would be the size of the font. You can also do Command plus to make the font bigger. So you can make it to whatever size is best for you. And Command minus to do it smaller. Yeah. Another thing is, of course, you can play around with all the options. You can go to settings, so terminal settings. And over here, you can pick colors and themes and all of that. So whatever makes your eyes comfortable, a lot of people don't like looking at a black screen, so they add some color. And sometimes the default screen is just white and that's too bright for you. You can change it to the dark theme. But yeah, this is something you can play around and find what works best for you.
Like that helps me be running a part of stuff.
How to have multiple, sorry, I didn't get.
Like, how we have like you run blah 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 blah.
Yeah.
So when you run something, the result shows that only your lines, lines, and then the result. If that's not what I say in my computer, like when there is like everything is like close together, so it's really hard to do.
Oh, really?
You know what I mean?
Can you maybe show me?
Yeah. So for example, like you like big and get them in the similar has. And then you look again. So everything is like together. But you have like a video of this.
Oh.
You know what I mean?
I feel just press enter and it time.
Oh, it was.
Yeah. Yeah. Because that's just a habit of mine. I like to continuously do enter to create these spaces. So yeah, I mean that's just how it is. But in the settings, I believe, so once you change your font, I'm not sure, but I think there is also like a spacing parameter. So you can change that, the distance from line to line. Okay. Yeah, so let's go back to Hoffman. And I'm assuming most of you transfer the folder to your home directory, correct? So in that case, you need to move it from your home directory into your Unix folder. So if you remember, that's the move command, the folder name, which should be like qcbio something, and source, and then the destination, your Unix folder. Okay. Okay, so you're good. Yeah. Yeah, that's only in case you transfer it to your home directory. Okay, you move it to Unix. Got it. Okay, yeah, that works. Yeah. Okay, now in your Unix folder, you should have this folder, which we got from git clone. So this should be uncompressed. And you should also have a zip file, the compressed version that you downloaded manually and uploaded with SCP or something like that. Right? Do all of you have the zip file? Oh, so you already unzip it? Yeah. Okay. Did anybody else unzip it already? You did? Okay. Yeah, so in this case, it's convenient because your MacBook already has an unzipping function. Um, for those of you that didn't unzip it, you can also unzip it from the command line. And it's fairly straightforward. Command is just unzip and followed by your file name. So it should be like qcbio dot zip. I think it's just zip, right? Now, there are different versions depending on the suffix that you have. So some files are .tar or .tar.gz. This is a different type of compression. And for that, you use the tar command. So tar to unzip or decompress tar files. Sometimes it's not .zip, sometimes it's .gzip. So we have also the ganzip function. Depending on the suffix that you have a different function that you can use. For a lot of biological data, you usually have what is called the block zipping, the block compression. So that one is usually like bgzip, but it's also .gz. Depending on the type of file that you have. Okay, so now let's go into this qcbio folder. Let's do cd qcbio. And you should see there is a PDF, there is a README, day 1, 2, 3, and slides. So just so you know, this is material for this workshop from older years. So if you need further material, you can always refer to that GitHub and get the older slides and material. Okay, so now that we're here, let's go to day 2 and list, and you should see these files over here. Okay, so yesterday we already talked about some of the commands that we can use to read these files. For example, we can do less, and then let's open this one over here, chromosome 110. and I can see the content of this file. These more specifically are the first few bases of chromosome 1. You see it's a bunch of ends. That's because the first few bases of the chromosomes are, well, they were unknown. They are known now, but yeah, this is one of the file formats that you work with in bioinformatics. This is what we call a FASTA file. And FASTA file just means you have sequences. In this case, you have the first few bases of chromosome 1. Okay, so we can visualize it with less. We can visualize it with more. Now, what if I only want to see the first few lines of this command? Oh, sorry, of this file. So there is a new function called head. And head by default is going to show you the first 10 lines, but you can always specify how many lines you want to see. So let's do, for example, the first 4 lines of this. So we do head -4, and we can look at the first 4 lines. Sometimes you don't want to check the beginning, but you want to check the end of the file. You want to see how it ends. We have the equivalent tail command. So tail, it will show you, tail -4 will show you the last 4 lines of the file. Right? So let's move on. So we talked about head, tails. Now we also have some other functions like zcat or zmore. So let's start with cat. Cat is, for most intents and purposes, the same as more. So this will also print all the content on your screen. As you can see over here, cat stands from concatenate. And one of the uses of this function is that it can help you to merge multiple documents together. For example, here we have chromosome 1 and chromosome 2 files. I can put them together into one. I can do cat chromosome 1 and then chromosome 2. And then I'll direct this to a new file. I'll call it chromosome 1, 2, and then FASTA. Now, if I open this new file, I see I have the content from the 2 files that I merged. So the function concatenate allows you to do a vertical concatenation. So put one file after the other. Okay. Yeah. So the new file that you create, it will have everything. But then when you visualize it, when you just open, this shows you one page at a time. Yeah. Okay, so in this directory, you see we also have a file that is compressed. Oh yeah. Oh. Yeah. In principle, you can name it anything you want. Here, I'm naming it .fa because it's a FASTA file. But yeah, you can name it whatever you want. Yeah. You don't have to, yeah. So one confusion I had when I was first learning about these things is that I thought that once you give a suffix to a file, then this file automatically has certain features, certain functions, and stuff like that. That's most of the time that's not the case. Like most of the time you can name a file anything you want. And in Unix, everything is a file. That's the philosophy of Unix. So it's gonna read it no matter what suffix you put. Now in computational biology, of course, you want to have a consistent format so you know what each file is. So it's more for self-consistency rather than function, really. Yeah. Okay, so that's the cat function. Now here we also have a compressed file, .gz. Now we have an equivalent of cat for files that are compressed and is scat. With scat, we can concatenate this to any other file that we have here. This is no longer a FASTA file, so I'm just gonna call it something else, like 10 text. Oh yeah, no, sorry. So for CCAT, we need both files to be compressed. So the second one is not compressed. So we're only gonna do the first one. And we should have our temporary file like this. Now, this zcat, it's also a way of decompressing because here we're printing the content of this compressed file into a regular file. And the regular file, we can just read it straight like that. So in this case, we're using zcat as a decompression tool. Now, this becomes a little confusing because it used to be the case that you needed zcat for compressed files and then cat for regular files. But then newer versions of Unix systems, like these functions work just the same for compressed or uncompressed. For example, less, it used to work only for uncompressed files, but now you can also do less and compressed files, and then you can read it the same. So, yeah, so these functions are becoming a little outdated, but just for your reference, there is an equivalent for compressed versus decompressed functions that are becoming more and more obsolete, but you have them there. Yeah. Yeah. Uh-huh. Right. Yeah. Yeah. So cat with one file is just printing that content. Cat with two files and then directing it to some output, it's merging them together. Yeah. Okay. So here is a question: does the order matter? And the answer is yes. Whatever you concatenate first is going to be at the top, followed by your second file, and so on. I think this became clear when we did this. So here we have chromosome 1 first, followed by chromosome 2. Oops. Okay. Another important tool, I guess another hack, if you want to think of it like that, is what we call a wildcard. Now, a wildcard is a character that you can use to replace other characters. For example, I'm gonna go back to the Unix folder and to our assignments folder. And here I have 4 files that all start with the letter with the word assignment. And I have 1 file that is different, right? That starts with 10. So what if I want to list all the files that have the name assignment? So I do ls assignment, and then after the word assignment, you can see it's different for each one of these files, right? I can use the wildcard. And this wildcard, the asterisk, will replace whatever string, whatever text follows assignment. So it's gonna list whatever file starts with assignment, dot, and then it doesn't matter what follows. So this is a way of finding files. Say if you have a directory that contains thousands of files, but you're looking for a specific type, you can use your wildcard. Now, there are different types of wildcards. The asterisk is the more general one. It will match whatever length of text comes after the word assignment. But what if I want to find my assignments 1 to 9 and skip assignments 10, 11, or whatever is greater? So I can do question mark dot text. Now, question mark is a wildcard for a given length. So in this case, it's only going to escape one character. So that's going to be assignment 1 through 9. Assignment 10, of course, that's 2 characters, is not going to match this anymore. So this will give me all the assignments that contain where the question mark can be replaced with 1 character. And then I match the end. Mm-hmm. Right. So files from 100 to 109. Or yeah. Right. Oh, I see. Yeah, so in that case, the number of characters is the same, right? But you're looking for a specific range. The easiest way I can think of is with a for loop. So you do something like this. So well, this is not part of this workshop, but you can write a for loop in Bash, and this is gonna give you a J variable that iterates from 100 to 109. And then you can use the J variable to write your file name. So for example, well, I don't want to confuse you guys further, but yeah, sure. Yeah, but just as a quick example, you will do something like this. So J will take values from 100 to 109, and then you replace the file name with the J, and then you can get all the files that you want. Yeah. Okay. Any other questions? Oh, this? Oh, you mean here I don't end it with anything, and here I end it with text? Or...
For example,
Mm-hmm.
first portion comes with that.
Yeah.
And then last portion.
Right.
And then what do
For this, yeah, anything of length 1. So 1 character. Yeah. Right. Yeah, so let's make an example. Let's say I'm gonna create assignment 11, assignment 12, and let's do assignment 102. So now I have all these new files, right? So if I do this, See, it's only gonna show me assignments 1, 2, 3, and 4 because these are 1 character in length. What if I want assignment 11 and 12? I can do this, or because I know they start with a 1, I can also do this. And if I want to do assignment 102, same thing. I can do it like this, or I can do 3 question marks. Yeah. Or if I don't know how was the length of the numbers and I just want all of them, I can also do like this. Asterisk will replace any length. Yeah. Okay. Okay, so let's do some practice. We're gonna look at this file over here. Let me go back to, right now I'm in assignments. I'll go back to Unix and then go to QC Bio day 2. And here we have this file, hg38 chrome or chr.fai. So what we're gonna do is, right now, let's just try search string by chromosome. Let's try some tools to visualize it. So we know less, we know more, we know head, tail. So just play around with those 4 functions using this file over here. And the other tasks, this slide is a little out of place. We're gonna complete the other tasks later on when we look at the other functions. So for now, just practice looking at this file with the different commands. So if we do less, we see we have 5 columns. We have 20, we have a lot more. We have multiple rows. And look at the first few lines. Let's see what are the last chromosomes over here. Okay, so quick question. Does anybody know what type of file this is? What this information represents? Sorry? Yeah, reference genome. Human reference genome version 38. This is an older version released in 2014. There are newer versions available. Here you have your different chromosomes. Here you have the length of each chromosome. Over here, this is it's a little harder to explain, but you can think of it as a cumulative length of chromosomes over here. Now, as you know, humans have 24 unique chromosomes or 23 pairs. But this file has a lot more than 23 or 24, right? Does anybody know what these fields are? yeah, they are contigs or scaffolds. So you can think of it as just different versions of these known chromosomes that are very common in the population, right? Yeah, that's not part of this workshop, but since most of us are doing biomedical research, I figure it'd be interesting. Now, what we're going to do with this file is we're going to apply some of our functions to do some file manipulation. The first thing we're going to do is, I mentioned there are way more than 24 chromosomes. How can we figure out exactly how many chromosomes we have in this file? Of course, you can always open the file and count, or you can do WC-L command. This command will count the number of lines in your file. WC stands for word count. Originally intended to count how many words you have in your file. But counting words is not always useful. The most useful application is counting total number of lines, because in most data formats, one line represents a data point. So yeah, so WC-L counts number of lines. Now, what if I want to, let's say I want to sort this file. Let's do sort. And by default, it will just print the sorted version onto your screen. Now, the default sorting is alphabetical. And one weird feature from Unix is that it will consider uppercase and lower cases like in different alphabets, so it won't mix them together. So in this case, we have the uppercase chromosomes first, sorted alphabetically, followed by your lowercase chromosomes. Okay, but maybe this kind of sorting is not that useful. What if I want to sort the chromosomes? Oh, yeah. Yeah, sort found. Oh, really? Who's it? Oh, really? Same. Who else has lowercase first? You do? All of them? You? Oh, so you did this. Do Control C. Oh yeah, that's another tip. Control C just kills the command and gives you a new line. Okay, so the sort function is really useful, and there is a lot of variations that you can try. So for example, one common one that you may want to do is something like this. If you do sort -K2, this means that you want to sort based on the second column. So it's going to sort based on column 2. Now, the default is alphabetical sorting. But column 2 is a number, right? We don't want to sort numbers alphabetically. So we can also further specify what kind of sorting we want. So we want to do column 2, and we want 2 to sort numerically. So we will write it like this. And then we put the name of our file. Okay. So now we have our chromosomes sorted from smaller from smaller to bigger. And here we have chromosome 1 being the largest, followed by chromosome 2, 3, and so on.
It's put into, you know, becoming to be happy.
Just the arrows up and down. Oh, Control C. Everybody has a sorted file. Okay. Is it sorted in this way for everyone? Yeah? Okay, good. Okay, what if I want to sort using multiple criteria? So say I want to sort based on column 2, but then if there is a tie in column 2, I want to break it using information from column 3, then you can just add more parameters. So first by column 2, followed by column 3. It should give you same because it's kind of already sorted that way. Here's the command. Okay, so we're looking at numerical sorting. That's n. What if I want? So if I don't put anything, that's alphabetical sorting. There is also lexicographical sorting. That means it's sort of an alphabet that includes letters and numbers, usually the numbers coming after the letters. So if you want to specify that lexicographical sorting, that would be a capital V. Of course, this only makes sense if we apply it to 1, because this here we just have numbers. Here we have chromosomes have letters and numbers, so we can apply this to this field. Now we can see we have our lexicographical sorting. And what if you have sort of like a mixture of values, or you have maybe some decimals or exponential notation? There is G. G stands for more general. Personally, I use G most of the time because it's sort of like a more intuitive sorting. So if you have a column that have integers like this, and then followed by some exponential value, and then over here maybe some decimal value, when you apply G, it will know how to sort, those values are in different formats. So it's a very smart sorting parameter when you put G, especially when working with numbers. Right, so we've done some counting. Let's do some, we've done some sorting. Let's do some cutting. Now, when we talk about cutting data, that means selecting specific columns. So let me do head HG38. This is our file, and it has 5 columns, right? Say I'm not interested in column 4 and 5. Like I don't even know what they represent. So I can do cut F1 to 3, 1-3, and then my file name. And this will give me the file with only the first 3 columns. So here's the command, 1 to 3. And at this point, here is some very important point is that when you're working with a command line, most of the files that you're going to work with are going to be tab separated. So what does that mean? If you look at this file, you can see there are these spaces between values. These are tabs. And most of the time people will use tabs to separate values because it gives you a consistent space between the columns. And when you run a function like cut, it's assuming your file is separated by tab. Now, it may not always be the case. For example, let me create a file real quick. Call it tempcvs 1, 2, 3, 4, 5, 6, 7, 8, 9. So this file is a comma separated value file. My values are separated by comma. So if I were to run cat on this, say 1 and 2, see, it's not gonna work. It's considering this whole thing to be one column, right? So the default behavior for cat is assuming your files are tab separated. What if they're not? So I'm gonna need to specify. How to specify? That's with -d and then comma. Now I'm telling cat that my values are separated by comma and I want the first two columns. So now I get the first two columns and still comma separated. So yeah, whenever you're using cut, just make sure your file is tab separated. Now, how to check if a file is tab separated? Visually, it's fairly easy. If you open the file and you see that the columns always start at the same position, so they are aligned to the left, then this is a pretty good indicator that is tab separated. Now, the tabs, depending on how your computer is formatted, its default is to 4 to 8 spaces. Now, what that means is that if your text is shorter than 4 or 8, then your tab is going to be a lot closer compared to the values where it's longer than 4 or 8. I don't know if that made sense. So it's pretty much marking a separation every 4 or 8 bases or 8 spaces. So if your value is longer than that, then it's going to jump to the next tab. In this case, here we have a value of 8. It's very short, so the next tab is going to be over here. So you can see the fourth column doesn't always align at the same place for all your values. Any questions? This is not sorted right now. This I just opened, yeah. Okay. So the cut function, you can just you can select whatever columns you want. Maybe a 1 column, 2 and 3, works the same way. Yeah, here we talked about the delimiter. This is the D function, F indicating the fields that you want. There is also an option C that if you want to select for the specific characters. This one doesn't really apply too much to what we're gonna do, but and it's replaced by what we're doing with capital D. So instead of comma, say you have values separated by a character, then you can also use your C. See what else? There is also another function called unique. So let's look at our file. Now, if we look at column 4 and 5, it looks like it's 60 and 61 for all of them, right? Now, how do we make sure it's always 60? What if this file has millions of lines and we don't want to scroll through all of them to make sure it's 60? So we can do, okay, we're going to do this. Now, there is quite a bit going on here, and I'm going to walk you through all of this. But for now, let me just type this. Okay, my output shows me that for column 4, there is 194 values, and with the value 60 and nothing else. So this way I can check that all the values are 60. But now here I did something weird, right? What are these lines over here? And why am I putting multiple functions that follow each other? This is a huge advantage of using the Unix command line, and this is called piping. And piping means that you can run multiple functions. You can think of it at the same time. So as a pipeline of functions. What I'm doing in this example is first I'm reading the file, then I'm cutting column number 4, and after that, I'm counting unique values. So I'm doing 3 different steps at once. Now, of course, here I split this into 2 different steps, but that is not necessary. I can directly cut. This will give me the 4th column. So cut the 4th column of this file. And then I put the pipe, which is just the straight, this straight line. And here I can do, okay, now that I have this information, I'm gonna run this function called unique-c, and this is gonna count unique occurrences. So unique occurrences of 60, 194 times, and there's no other value. Okay. Yeah, so it tells you 194 lines have 60 in there. Right. Yeah, exactly. Yeah. Third column. Uh-huh. Yeah, so depends what you want to check, right? So if we look at the third column, we see the numbers are different, but we don't know for sure that the numbers are always different, right? So we could do the same thing, unique C. And yeah, in this case, well, in this case, it's not super useful because we have a different number for every line. But it looks like every number is unique. Every number appears one time. Yeah. Or more, yeah. Mhm.
For example, we have 196.
Yeah.
But only like a 1600.
Right.
But you have fast, very fast. You have to.
Yeah, exactly. So for example, so we know column 4 is the same, right? But what if I go to my Vim editor and I manually alter the data? I'm gonna put a 62 here, and here I'm gonna put a 65. Okay, so now let's do the same thing. This is column 4, piping, getting unique elements. Okay, so Now we see there is 162, followed by 460s, followed by 165, followed by 60. So it does give you unique elements, but it respects the order. So it didn't merge these 460s with these 188 60s, right? So it is a little tricky. Now, what can I do if I don't want this? I want just unique values. I don't care in what order they appear. What should I do? Because let me show you the file. Right now, I have 62, followed by 1, 2, 3, 4, 5 60s, 165, and then everything else is 60, right? And here is my 62, 60, 65, and then everything else is 60. What if I just want to get the count of unique values? I don't care about the order. What should I do? Yeah. Exactly. Yeah. So this is where the piping commands come in place. I cut the fourth column. I can sort it. And then I can do unique C. Now I have all the unique values, the counts of all the unique values. Okay. Is 3:25. I think we can take a 10 minutes break now, and we'll continue at 3:35. Okay. Any questions so far? Okay. So we had a very quick intro to piping, but yeah, once again, this is a way that makes Unix a very practical language that you can just combine all of these different functions at once. So we know, for example, sorting, we know cutting, we know how to count lines. We can do something like this. So let's look at our file again. So here we have chromosome names. We have the length, positions, and then the 60, 61. Let's look at the fifth column. So let's cut the fifth column. Now, what can we do with this information? Why don't we count how many lines it has? So of course, it should be the same number of lines as the whole document, 194. What if we want to count unique lines? How do we do that? We actually can use the sort function -u. So sort -u, it will sort the values, but if there is a repetition, it will only show one of them. So we can cut the fifth column, remove duplicates, and then count the number of lines. See, now we only have one line. So this is also another way to check that column 5 has one unique value that repeats 194 times for all the entries, for all the chromosomes. Right. WC-, yeah, per line. Unique, yeah. Okay. Now another example, I cut column 5. Well, let's do something else. Let's do column 2. It's more interesting. And okay, but I don't want all of column 2. I just want the first few values. So I can cut and then do head. So this will show me the first 10 values. And here I have the first 10 values of column 2. And now I want to sort. I'm gonna do sort. So instead of sorting the entire file with this piping command, I'm only sorting the first 10 values of my file. So you can already see how with these piping commands, you can do all weird combinations of functions and commands to manipulate your file. Let's look at another example. Okay, let's start with, what if I only want to work with the canonical chromosomes? So what are the canonical chromosomes? Are the ones that start with CHR, right? So we have our canonical chromosomes, and then we have all these other weird scaffold contigs, right? So what if we only want to sort our canonical chromosomes? So I know we have 25. So I can start by showing the first 25 rows. So I'll do head 25. And now I'm going to sort them based on their length. Okay, so first 25 rows, type, sort by the second column. And now I have my chromosomes from shortest to longest, starting with the mitochondrial chromosome all the way to chromosome 1. Yeah. That has to do with the tab. So let me show you an example. I'm going to create a new file, temp.tsv. TSV stands for tab separated values. I'm going to write one, say this is just a list of name and last names, right? So I start with one name and one last name. So I'm writing the name, I press tab. See, it jumps to the next tab, right? And here I write the last name. Say the second name I want to type is very long, like my name, for example. Well, let's say my name is even longer. Now, when I press tab, you see, it's not going to align with the previous last name because it overlaps, right? So, yeah, so that's why even though they are tab separated, they may not necessarily align because of the length of the values. Yeah. Okay. Any other question? Okay, so we're learning about piping. And yesterday, I also introduced this one command, the arrow, right? Yeah.
Dot AG on the TN. Because it's
Uh,
just a column T.
Yeah.
And you in that specified column T.
Yes. So if I do like this, it will sort based on column 2, but the default is alphabetical sorting. Yeah. And because this is number, we want to do numerical sorting. Yeah. So that's why we added the comma to it. Any other question? Okay. So I was saying that yesterday I introduced also this command over here, the arrow, and this is to direct the output, right? So over here, we're selecting the canonical chromosomes and sorting them. I can direct this to a new file. So I'm gonna call it hg38 canon only dot fai. And when I open this should have my output, right? Here we have only canonical chromosomes. I can check by counting the number of lines. And here we have 25. Okay. Now say, what if I want to add something to this file? Say I forgot to select. Let's think of this hypothetical case that there are actually a few more canonical chromosomes that I didn't select here and I want to add. How can I do that? So let's open the file and let's say I want to add this last few chromosomes to my canonical chromosome files. So I'll do tail 4. So this will show me the last 4 chromosomes. And now if I want to append to an existing file, I will use this double arrow. I'll copy this. And now I can check. Okay, my new file has the canonical chromosomes, and it also has these additional ones I just added. And if I count the number of lines, okay, now I have 29 instead of 25. So one arrow is to direct output. If the file doesn't exist, it will create the file. And 2 arrows means appending. So the file must exist beforehand. Well, actually, it will still work if it doesn't exist, but the point of the 2 arrows is to append to an existing file. I think that's better worded. Okay, so far we already have quite a few tools to do some file manipulation. Now, if we go back to our slides, we have this symbol here. This is the piping symbol, so the straight arrow and this redirecting symbols. Now, let's look at grep. Grep is a very useful function that helps you to find strings in your file. So for example, let's open this file again. And let's look at these weird chromosomes. You see a lot of them, they start with GL000, right? And we have a lot of them that follow this pattern. And we have some other that are KI270 plus some number. So what if I ask you, okay, create a new file that only has the GL00 pattern? So what we can do is I'm gonna open pipe grep, and here I'm gonna write the pattern I want to find. GL00, I think it was 3 zeros. 000. And this will find all the chromosomes that have this pattern. And now I can direct this to SG38 GL chromosomes. .fai. Okay. So grep is one of the most useful tools that we have because it allows you to subset your file very easily. Here in this example, we're selecting the GL00 chromosomes. But what if I ask you select all the chromosomes except the GL00, so the reverse task? For that, we can use grep-v. This -v does the reverse search. And I can direct this to non-GL chromes. And if I open this file, we can see I have all the chromosomes except the GL chromosomes. Okay. So that's one scenario. Say, for example, you want to exclude a string, or you want to find a string, but you don't remember if it was written in capital GL, in uppercase GL, or lowercase GL. So what you can do is -I. That means ignore the casing. So it works if it's uppercase or lowercase. And of course, you're probably thinking, how am I going to remember all these options, the -I, -B? That's the thing, you don't have to remember them. You can always do grep --help, and you can look at the documentation. So -I, ignore case. Let's find -B, invert match. We see there are some other options like -F, for example, obtain a pattern from a file. -W, where it has to match the entire word. So it doesn't do substring matching, but it does like word by word matching. Yeah, we have, and you can also control the output. For my daily use, at least the ones I've used the most, and I think what most users use the most is the -I, -B, -W. So let's do an example with -W. If we do -W GL000, we see we don't get anything, right? Because W is an entire word, is word by word matching. So this is a substring. This only matches the beginning of the word. So it's not going to give you anything. But if I do the entire word, so for example, I know there is a chromosome 7, they should give me one match. Yeah, so, and -F is also good. Here we can do something more advanced. So for example, I already have a file that has my canonical chromosomes, right? So, well, canonical plus 4 weird ones. Okay, so I want to find these chromosomes in my initial file. So how would I go about it? I know they are in column 1. So I'll cat column 1. I can always type and just see this intermediate output. So cat F1 less. So here I see I only have column 1, so I have the chromosome names. And I can redirect this to a temporary file, which I'm gonna call chrome names. Okay, now from the bigger file, so this file has everything, right? It has 194 chromosomes. I only want to find the canonical chromosomes that I saved in these chrome name files. So I can do less pipe grep F chrome names. So it's going to match whatever is in this file and search for those matches into this bigger file with -F. Yeah, it's quite a lot of information. I'm throwing at you now. So let's try to summarize. Grep, in general, helps you find a string within a file. Grep V is the opposite, everything but the string. Grep I ignores case, so it finds uppercase and lowercase of your pattern. Grep W, word by word match, so it has to match the entire word. Grep F, it's the file match, so it will find all the patterns that you wrote in a specific file. And if you ever forget, grep help, and you can find the description. Okay. Any questions? Would you be confident if we had the quiz right now? This part. The grep feels a bit more complicated. Okay. Yeah, so let's do some exercises then to practice this a bit more. Let's open hg38.fa. Okay, how do I select all the chromosomes that have GL00? What do I do? So I already wrote this. Now what should I write next? Pipe, yeah. Grep. And what? Sure. And the pattern I'm interested in, right? Yeah, and now that gives me all the matches. And what if I want to save these matches somewhere? Arrow. New file. Yeah. GL chromosomes. Okay. What if I want everything but the GL chromosomes? Dash B. Yeah. Yeah. And I can save it in the non-GL chromosomes. Okay. What if I want to do an exact match for chromosome 1? Actually, this is a good example because let's say I do this. Okay, I should clarify also, when you grab a pattern, you don't have to put it in quotation marks. Like it will work if you put it without the quotation marks. But it's a good habit to put the quotation marks because there are like some special symbols and patterns that you absolutely have to put them in quotation. So it's good that just in general, you always put it. Okay, what if I do like this? I want to find chromosome 1, so I do grep chromosome 1. It gives me chromosome 1, but it also gives me chromosome 10, 11, and you can see why, because it also matches, right? Exactly. So if I'm interested in only chromosome 1, I have to use dash W. Yeah.
You don't specify the polygon
Uh-huh.
if they happen to be from some 1911 area, it will come to.
Yeah, that's a good question. In general, it will show you any line that has your pattern, regardless of the column. Yeah. Now, when we talk about awk, this is a bit more complex, but we can you can specify, like limit it to these specific columns. But for grep is more general behavior. Yeah. Okay. Any questions?
So that's also use like big sets same thing that the driver service, but.
Dash W, yeah. It has to be it's word by word match, yeah. Okay, and yeah, the last one that we did, the grep F, is a bit more sophisticated because if you look at this, we're using grep followed by a string, right? That we want to search. But when we do grep F, we follow it with a file name, like Chrome names. So it's different in that sense. Grep F followed by a file name. Now, this is useful when you're interested in finding, in this case, all the GLs have a clear pattern. They all start with GL00. The canonical chromosomes, they have a pattern. They all start with CHR. So it's very easy to grab and find them, right? But sometimes you may be looking for a set of things that don't have a clear pattern. But you can always write them in a list. One example where I remember using this is you can have a file that's called like genes of interest. And this is probably a file that you downloaded from a paper, like some supplementary file. And it has like a list of genes that you are interested for your research, right? And now you have a file that, well, actually, let me show you one of my own files. Data. Okay, so this is a file that has information about all the genes in the human genome. So you can imagine, you can see that it has a lot of columns. It probably has several thousands of rows. Let's count. So just counting is going to take a while because this file is just so large. But if I want to find information on my genes of interest that I wrote them here, then I can do grep F genes of interest. And see, it's only gonna show me the lines that contains these genes. FXR1 is here. FOXP is over here. It is controller. Yeah, that's exactly what it is. Okay.
That's another thing to be able to listen to
Exactly.
that's the way.
Yeah, that's a great way of explaining it. The equivalent would be to do grep W and then going gene by gene. Yeah, that would be equivalent. And then appending to the same file. Because this will give me one output, then FOXP will give me another output, and then I have to merge them.
So then you grab that from everyone find this it works.
Yeah. You have to do it individually. There are ways of grabbing multiple patterns, but it gets confusing. So it's usually recommended that you either put them in a file or you do the individual searches. Yeah. Okay. And here. How do they get highlighted, you mean? So when you do grep, it's gonna highlight the patterns that you are searching for. Yeah. Yeah, it's not highlighted in the original file. Uh-huh. So the way it works is it's going to show you the entire line that contains the pattern and only highlight the pattern itself. Yeah. Okay. Any other question? Are we good with grep? At least better? Okay. Shall we move on or do we still have questions about grep?
Lastly.
Uh-huh.
Grab that.
Yeah.
Is it, I'm guessing it's gonna separate each one by like the.
Actually, no. In this case, it's expecting one turn per line. So this is something, yeah, I didn't specify, but yeah. Here you should have one word or one pattern per line. Yeah. Yeah. Any other question? Okay. So we have piping, we have grep. Now let's do awk. Now awk, it's yeah. What time we end? Oh, 4:30. Yeah. Okay. Okay, so grep is a command that it lets you parse, it lets you do some bit of manipulation of the file line by line. That's the main idea of awk. So right now we can do things like cutting, subsetting based on the number of rows with head or tail. We can select specific columns with cut. But if we want to do something line by line, then awk is the tool that, more easily we can do that. So let's start with an example. Let's say I start reading my file. Okay. Now I'm gonna type, I'm gonna do awk. And the format is a little weird, but you have to start with the word awk, quotation marks. So this is one of the cases where the type of quotation matter. You have to use the single quotations. And the brackets matter. You have to use the curly brackets. The spaces don't matter. Oh, sorry, this space matters. Yeah, this one between awk and the quotation, yeah. But then inside, these spaces don't matter too much. Okay. And now we can do an operation line by line. So for example, if we do print $1, this means print the first column, the first value of that line. Now we pipe it and we do less. And we see now we only have the first column. So far, this is equivalent to just cut F1 and then gives you the first column. So far, it's the same thing, right? I can also select multiple columns. So print $1, $2. Now I have column 1 and column 2. Okay. What if I want to do a slight modification? So say, for example, I want to add a little string before the chromosome name. So I want to do something like, I want to add this symbol before the chromosome name. So I can add the symbol, in double quotations, then followed by the chromosome name or column 1 and then column 2. And now I have added this. So you can already see how this awk lets you do file manipulation more easily. Let's see another example. I only want to see chromosomes that are longer than a certain length. So I don't care too much about these shorter chromosomes. I can add conditional statements. So for example, I type if parenthesis, column 2 was the length. If column 2 is greater than, let's say, 1,000,000, then print the first column, column 1. And now it's only going to show me the chromosomes that are longer than 1,000,000. Okay. So there's quite a bit going on over here. So let me break it down again. Here we're using awk inside of the piping. And whatever we put inside of this quotation and bracket is an operation, is a function that we can run for every line by line. Over here, we're just printing, so that's fairly straightforward. Here, we're adding a character. And over here, we're doing a conditional subsetting. So before, our subsetting was just limited, like the first 25 lines or the first n lines, and then we subset into a new file. Now we can do something more interesting, like, okay, we only want chromosomes that are longer than this length. So we can add a condition like this. Any questions? I know this feels like now we're jumping into yet another level of difficulty, right? And it gets a bit more confusing because awk has its own syntax that is a little different from Bash, from the rest of Bash. So inside of, for example, here we're writing an if statement like this. If you want to write an if statement for Bash, it's actually written in a different way. You don't write it like this. So you can think of this as a very quick and dirty programming tip that will allow you to do some file formatting like while you're piping. Now, let's look at more cases. For example, here I'm printing the first column. So it's just telling me the chromosome names of the chromosomes that are longer than 1,000,000. Okay, what if the chromosome is longer than 1,000,000, then show me the entire row. So if I do $0, this shows you the entire row, not just a specific column. And it gives you back your original file. Okay. Is there any questions? Is it clear? Can we add awk to the quiz? No. Okay. Let's look at more examples. Let's see. Sorry? Which one? This one? So first Q. For the hour. Oh, you're meeting on. Yeah. Does it don't work to be done for you? Is it working for everyone? Yeah. Where are these songs? Yeah, or you can also try rerouting it. So maybe you know something the first time. Cut. Okay, so awk lets you do all these operations really efficiently. Another application of awk is that you can also do like some operations. So for example, let me look at the first, I'm just gonna print it again just to see, oops. Just to see like the first few rows. Okay, so column 4 and 5 are probably meaningless, but say I want to add them. So I want a new column that is the sum of these two columns over here. So I can also do that very easily on awk. So I'll do print 0. So that means print all the original columns. And I can also do column 4 plus column 5. And instead of less, I'm going to do head. Okay. So now you can see we have a new column that is the sum from these previous two columns over here. So this is one clear advantage where you can do operations. So you can also further process your file. We can subtract as well. So we get 1, -1. And another very useful operation as well is running sums. So for example, what if I want to do the sum of all the values on column 4? So that one we can do like this. I define a variable n that sums column 4 and then print n. Now this is a little different than the other command. So let me break it down. This first part is the standard awk that we talked about. It goes line by line. So for every line, we're summing the value on column 4 to this variable n. Then we're hitting the end of the file. And now I'm just asking, okay, show me n. Show me what is the total sum. And it will give me the total sum of all these values. Now, let me recap this. We have 6 minutes left, so let me recap the awk examples that we may have to deal with in the quiz. So the first one we can use awk to show specific columns, right? So column 3, it will be like this. If I want to show column 1, like this. And if I want to show all the columns, I put 0. Okay. So we're good with that one, right? Great. Another case would be if we want to add a string or add some text, add a new field into our file. So we talked about adding this element at the beginning. Now we have this in all the rows. This I didn't mention, but I can mention it now. We can add an entire new row if we just put a comma. So new, and then the original rows. So we have a new row and the original ones. Okay. So new row, and if I don't put the comma, it's just gonna be together. Okay. Are we good with that? Great. Another common case is subsetting, conditional subsetting. So the way it works is if parentheses, inside the parentheses, you put your condition, and then print the lines that pass that condition. And the example we looked at was if column 2 is bigger than 1 million, then we print all the columns. Okay. So conditional formatting. This one is a little bit more complex. So you have to remember if parentheses print. Okay. Now, yeah, the last case was how to get, oh no, there are 2 more cases. So what if I want to add the values? So I can print the original columns and then add the values from column 4 and column 5. So that gives me my original 5 columns and a new column with the sum. So we can do this operation, sum, multiplication, division, rest, subtraction. Yeah. No, so if we do that, it's only gonna show you that last column. It's only gonna show you the total. Okay. Right, yeah. So most of the time, well, at least when I use the command line, it's useful to add more information to your existing file so that your file is more complete, more comprehensive. Okay, so column operations. And the last one is if I ask you to count what is the row sums for column 4. So this one is also a little more tricky. We start with 1 bracket, then end, and then another bracket. Here we define a variable. So we're summing the values of column 4 to this new variable R. We iterate all the columns, we end, and then we print R. Yeah.
So in the 3 examples, it seems like you did multiple things within C of bracket. Why is it different that we have to have?
Yeah, that's a good question. So we said that awk does something line by line, right? So here we only have 1 bracket. And for every line that is going at the time, it's just printing the original columns plus the sum. So it just, once it goes line by line, that's the entire task. It doesn't need to do anything else. Here is a little different because it's going line by line, summing this value to R, but then it has to end before showing you what R is. In this case, R is a sum of all the rows. Whereas here we're doing something row by row. Yeah.
Is it possible? With the same number, because I have 1164, sorry, 11,603.
Yeah. Remember I manually edited some numbers. Yeah. So you should get slightly different. Yeah. Uh-huh. Oh, you mean this? Yeah. So in general, this means a cumulative sum. So plus equals. It just means you keep summing into that R variable. Because if I did this, for example, R equals, so R is getting changed in every row. And it's just going to show me the last value of R. If I do just plus, it's probably going to give me an error or nothing. Yeah. Because it's not meaningful. Yeah, so that would be like asterisk equals if you zero. Maybe I have to, I'm not sure. I'll need to look it up. The thing is that also it's probably a really large number, so I don't know if it's gonna show. Yeah. No, so R is gonna get redefined every time you open awk. So you can think of the life of R just limited to this command. So it doesn't matter if previously was zero, it's not gonna be zero again if you run awk again. Or are you saying that there is a zero somewhere? So no. Okay, okay. Right. Okay, yeah, so we should end the session here. There is still some more tools like set that we didn't cover today. So don't worry, I'm not going to include that in the quiz, but I am going to include everything until awk for tomorrow. Of course, if you still have questions, we can always review a bit more before the quiz. Okay. Yeah, that's it for today.